Hi everyone, my name is Minjun Park from Baylor College of Medicine. Today, I'll be presenting multinomial convolutional neural networks for mechanistic modeling of enhancer activities. One fundamental question that we ask in regulatory genomics is how are the genes regulated? And it has been found that regulatory sequences, also known as enhancers, recruit regulatory proteins, also known as transcription factors, that regulate gene expression. Here, an enhancer is a short DNA region where transcription factors bind to activate or repress gene expression. This description of gene expression regulation gives rise to two mechanistic questions. First, which locations in an enhancer do transcription factors target for binding? Secondly, how are the regulatory effects of enhancer-bound transcription factors integrated toward regulating a gene's expression level? State-of-the-art approaches that answer these questions do not provide mechanistic understandings. They utilize a broad collection of sequence-based features and epigenetic features, such as GC content, evolutionary conservation, and the existence of histone modification in the sequences being modeled. However, these features do not explain where TFs bind and how the effects of transcription factors integrate toward gene expression. They also deploy deep neural networks that are difficult to interpret. These models need post-hoc interpretation, and it is unclear if a biophysical model could directly use those post-hoc discovery interpretations and achieve similar accuracy. To overcome these challenges, we propose multinomial convolutional neural networks where convolutions model multinomial distributions that are directly interpretable as classical sequence motifs of TF-DNA binding specificity. A multinomial convolutional neural network is broadly applicable to any dataset of sequence to quantitative scores. We use the acronym MUSEUM for our model. It means multinomial convolutions for joint modeling of sequence motifs and enhancer activities. Now, I'll talk about the architecture of MUSEUM. MUSEUM first takes one-hot encoded sequences as input. In the convolution operations, MUSEUM learns convolutions as multinomial distributions over the four nucleotides. With show matrix operation, MUSEUM transforms one-hot encoded sequences into likelihood ratios between the multinomial distributions and a background distribution. Here, the multinomial convolutions are directly interpretable as motifs of TF-DNA binding specificity that has been classically used in biophysical modeling of genomic sequences. In the max pooling layer, MUSEUM chooses the highest likelihood ratio, that is, MUSEUM selects the strongest binding site for each model motif. Then, we use a linear regression model to fit the sequence's enhancer activity data. Here, the coefficients represent the regulatory effects and strength of the transcription factors. As proof of principle, we apply MUSEUM on a recent, high-quality, massively parallel reported assay dataset of more than 2,000 human genomic sequences assayed in the human liver cell line. MUSEUM achieved high performance of Spearman correlation coefficient of 0.54 in tenfold cross-validation. The multinomial convolutions in MUSEUM identify motifs of TFs that have known function in the human liver. The coefficients of these motifs in the linear regression model also match with the known activating and repressive roles of the corresponding TFs. For example, MUSEUM identifies ATF1, the activating transcription factor 1, as the strongest activator. ATF1 is known to play an activating role in the human liver. Finally, we check if MUSEUM motifs pairs have captured any co-binding patterns known for regulatory TFs. Interestingly, about 21% of MUSEUM motifs pairs indeed capture the formation of known TF complexes. MUSEUM also showed a high generalization. When we applied the model to predict enhancer activity of DNA-seq peaks in the human liver cell line, MUSEUM accurately predicted the decrease in activity with decreasing accessibility. Furthermore, MUSEUM predicts high levels of activity for those peaks annotated as enhancers and transcription start sites, and predicts the low levels of activity for the repressed regions and weak enhancers. We also used MUSEUM to prioritize functional single nucleotide polymorphisms, or SNPs. We define an SNP's predictive variant effect as the change in enhancer activity of the sequence flanking the reference allele when it is substituted by the alternate allele. At 5% FDR, MUSEUM prioritized 416 common SNPs as potentially functional. These SNPs are significantly enriched for low minor allele frequency and commonly occur in regulatory sequences. Overall, MUSEUM achieves high performance with high mechanistic interpretability, a feat not realized by other state-of-the-art models. The model can also combine more complex architectures while retaining full interpretability of the multinomial convolutions. Thus, our approach will be transformative in building more interpretable models of genomic sequences. Here are our funding resources, and thank you very much for listening.